I'm one of the Pharmacology Toxicology supervisors in the Office of Oncologic Diseases in CDER, and I'm going to talk about getting to first-in-human trials for small molecules and biologics. I'll start with an overview of who is the team when you submit your IND, where to start when you're looking for how to submit the IND, what we're looking for, and a couple of case studies for your consideration. Some of the abbreviations that I'll use in this talk include the HNSTD, which is the highest non-severely toxic dose, the STD-10, which is the severely toxic dose in 10% of the animals, mechanism of action is MOA, POC is proof of concept, the minimal anticipated biological effect level, or MABEL, GLP is good laboratory practice, SEND is the standard for exchange of non-clinical data, ICH is the International Council on Harmonization, and FIH is first in human. Usually start with your initial team. So when your IND comes in, you're assigned to a team. You have a project manager, which is your contact person, does all of the regulatory process work, and is really your communications person. You'll also have a CMC review team. They look at your manufacturing, your process, your impurities, your batch data, your stability, etc. ClinPharm is assigned very early on. Even though you don't actually have any clinical data, you'll make comments from them about food effects, drug-drug interactions, your clinical PPK plan, and your anti-drug antibody assessments. And then you'll have your clinical reviewer who's looking at study design, your DLT definitions, your stopping rules, and your patient population. You may also have a statistics reviewer or a device reviewer, depending on what kind of data and what kind of package that you have. But your primary pharmacology toxicology person is probably the person reviewing the most data with that initial IND. The PharmTox review team looks at the mechanism of action and proof of concept, sometimes metabolic comparisons to help support your species selection for your tox study, all of your safety data, and really determines whether or not you have sufficient data to support your proposed starting dose. Non-clinical, because of this, is very front-loaded. So if you have a paper file, which we don't have anymore, but originally when I started, we had a lot of paper files. And what you would see when it came in is that you have a little bit of clinical data because you have your clinical protocol, obviously, and they're also going to look at your informed consent document in a lot of cases. You have CMC data because you have an actual batch that you're looking at, so you have how it's made and the process. But the bulk of your initial data for an IND is going to be your PharmTox data. And you've had your great idea now. So what do you do next? You've identified your target. You've shown that your drug has an effect on that target. You've shown activity in an in vivo tumor model. Maybe you have some xenograft data. Mostly people have xenograft data to support oncology drugs. You don't have to necessarily have a xenograft model for every tumor that you might attack, but we understand that you use the xenograft data to help you in some of your planning. Sometimes people will use it to help support whether they want to do single doses or they want to do BID dosing, and also in combinations. If they want to support combination drug development very early, you'll have xenograft data to help you do that. You can incorporate some safety endpoints in this data, like body weight loss, mortality. Sometimes people will include PK depending on what they're looking for, or they'll take out the tumor and look for biomarker data. All of this data is very important, but it's not necessarily safety data. So basically that is not enough for your in vivo tumor model. So what's next? Well, you have to start with the guidances. Dr. Layton yesterday, I believe, gave an in-depth discussion on ICES9 and how it was developed and the differences for oncology drug development versus development of drugs in many other therapeutic areas. Also, the ICES9 Q&A document has a lot of good information. ICES6 talks about biotechnology-derived products, so a lot of the antibodies, and it also has an addendum. And I've also included here the guidance for estimating safe starting doses. This is for healthy volunteers. We don't necessarily use the same endpoints because we'll use an STD-10 or an HNSTD instead of a NOAEL for our start dose recommendations, but the overall process is very similar, and it can be a useful place to start. So, as I said, ICES9 explains the basic expectations for anti-cancer drugs. Usually you have 28-day GLP-compliant toxicologies in 2 species to support the development of a small molecule. For biologics, you'll have a single GLP study in a pharmacologically relevant species because often there's not an acceptable or pharmacologically relevant second species. And these studies are the primary data used to determine the starting dose for first-in-human trials, with the exception of some of the immune targeting drugs, which ICES9 does call out an exception on the standard toxicology-based methods of the starting dose for some of these immune agonists. And you can use a MABEL to support those starting doses. And the MABEL relies heavily on pharmacology data. So what are the endpoints in these toxicology studies? Well, a good place to start is the SBIA Red Book. It gives some detailed descriptions of toxicological endpoints to include, and it has a large array of study types. It ranges from gene tox to chronic tox. And I have this link included here. Some of the key endpoints in a GLP toxicology study include mortality, clinical observations, body weight, food consumption, ECG, ophthalmology, hematology, clinical chemistry, gross pathology, organ weights, histopathology, and toxicokinetics. Safety pharmacology endpoints are usually built into toxicology studies for oncology drugs as well. And they would be both in rodents and non-rodents. So in rodents, sometimes you see a functional observational battery or Irwin test built into your tox study. Also, you look for stereotypic behaviors. Those can sort of suggest that you may have some CNS liability. You'll look for wheezing or gasping, or you can do plasmography studies that are included in your rodent study. You look for some of the same clinical observations in your non-rodent study, but your non-rodent study usually also includes an ECG assessment. Maybe on day 1, maybe later. Usually there's only one assessment. You could do 2 to try to get a sense of whether there are immediate acute ECG effects, which is what you would see in a standalone cardiovascular study, or looking for longer-term effects. Cardiovascular endpoints are also assessed sort of in an in vitro hERG assay. So that safety pharmacology study is something that wouldn't be built into a GLP tox study, but is often included as well in a non-clinical package for an oncology drug. So what are some of the stumbling blocks for your initial IND? Well, one of the ones are the intersections of PharmTox and CMC. The big ones being not having a low enough dose formulation to support your supported starting dose, not having data to showing that you can deliver the right dose, or using a tox batch that is too clean, because we don't have to have exactly the same impurity profile as your clinical batch, but we want to make sure that the clinical batch, the tox batch, is representative of the clinical batch, and that if there are any major impurities at high levels, that they have some coverage, at least at the starting dose, to support development. Another stumbling block can be just GLP itself, and one of those things would be a signed pathology report. Pathology reports under GLP are considered primary data and therefore must be signed to be considered valid. Another stumbling block may be SEND data, which is the standard exchange for non-clinical data. This is a fairly new requirement that's going to become more of an issue, because with certain toxicology studies, if these data sets aren't included, the technical rejection criteria is going to be turned on and your IND may be rejected at the gateway, so that we don't even know that it's been submitted. So what are good laboratory practices? GLP stands for good laboratory practice, but this doesn't just mean a study with good controls and endpoints. It's not an assessment of how good your lab is, necessarily. GLP is these things. You do need the right controls and inclusion of the right endpoints, But it's also details about archiving, study conduct, and responsibility for different pieces of the study that allows for inspection later on if there are questions. GLP is described in the Code of Federal Regulations, 21 CFR Part 58, that's shown here. And it's a big part of whether or not we can actually accept your study. So some points about these GLP studies: you need to consider your CRO or your conducting lab. The studies are expensive. You want to make sure they're done right. The studies are also foundational, and a bad study may not save you money. So make sure that, and also make sure that they can format the data and send, because that's going to be a big issue going forward. So what else do you need? You have your pharmacology data, you have your toxicology data. Is that all? Do you need more? Well, it kind of depends on your product. Screening data can be important, looking for other targets. So say you have a kinase inhibitor that you're using to inhibit EGFR, and you've shown all of your studies that it inhibits EGFR really well. But you also need to show that it doesn't also inhibit VEGFR really well. We're looking for that early on. Also the secondary pharmacology. So these might include screenings looking for opioid receptors and GPCR interactions, because for your small molecule drugs particularly, you can have these off-target effects. Another thing that we like to see is concentration response data. So you want to have an IC50 that you need more than one concentration that you've used. And a xenograft study that only has one dose that has been studied is maybe less valuable than a xenograft study that has a couple of doses, so that you can see if there's a difference in the response. Another thing that people get tripped up on, especially for the immuno-oncology products, is the cytokine release assays. These are hazard ID studies. We're looking for potential for acute cytokine reactions, both in soluble and plate-bound formats using primary human cells. And this lack of this data for some of the IO products has resulted in at least temporary clinical holds. And then the concentration response data is also important for these IO products, because it can really inform on starting dose, given that you have to use a MABEL for a lot of these products. And then the safety pharmacology, which we've discussed earlier. The in vivo endpoints are usually built into the tox studies, but the in vitro hERG assay may also be included. Later on in development, phototoxicity and gene tox are going to be two of your big things to consider, especially for small molecules. The gene tox you usually don't need for oncology drugs until you support your marketing application, unless you are trying to include healthy volunteers in any of your studies, in which case you need to generate that gene tox data earlier. Phototox is another thing that's often done early. That's because you want to inform on whether people need to be protected from the sun very early in their trials. And some of these are in vitro data can inform on this, and there are some standard assays like the 3T3 assay that are used to assess phototoxicity. So if you've done all of this and you've looked at these guidances and you still have questions, you can have a pre-IND meeting. And pre-IND meetings can occur at different stages of development. Some of them happen very early in development, before the tox studies are done, and some of them happen later, when most of the questions are really about clinical study design. And you really only get one official pre-IND meeting, but you can request additional kinds of meetings. Most of these are going to be written response only, but we will try to answer your questions. Some of the pre-IND considerations might be to provide enough data that we can really answer the question, but understand that we can't pre-review all of your primary data. So when you ask us, is my start dose supported? We may tell you if we think that it clearly isn't supported, but we're not going to agree to a start dose right away. The other thing to consider is that we're not going to tell you exactly which pharmacology studies to run. We might say that we're not sure that you've really supported your mechanism. We might say that there's some issue, if we see a specific issue that doesn't seem supportive of what you're trying to prove, we may bring it up, but we're not going to tell you exactly which assays to run. That is part of your development process and something that you have to decide. And something to keep in mind is that we will put a drug on hold for safety reasons, but we will not usually put a drug on hold because we're not sure that we agree that it's going to work. I mean, this is an experiment, really. So we could be wrong, so we're not going to put you on hold for that, but we will point out some of the big issues if we see them. Another thing is to not ask really vague questions like, is my protocol okay? That's too non-specific. You're not going to get a very useful answer from us on something like that. And now I'm going to give you just a couple of case examples to help illustrate some of the points I've made. So case one is SMKI-152, which is a small molecule kinase inhibitor. The intended primary target is not novel, and it's not first in human, not first in class, but it is first in human. And it has several other targets identified from a 500 kinase panel screen, but no additional hits on secondary targets. They did toxicology studies on 2 species, the rat and the dog. Their histopathology reports were unsigned, but they had an STD-10 of 100 mg per kg and an HNSTD of 6 mg per kg with a proposed starting dose of 75 mg. So, some of the issues for SMK-152. Issue 1 is the unsigned pathology reports. This is a GLP violation, and those reports are not considered valid. So the first thing we did was to send an information request asking for the signed report. That is always going to be our first step. We're not going to just throw your study out, but we are going to ask for an update and see if you can provide the additional data. The sponsor in this case was able to provide the report. The problem was resolved. Issue 2, which is a more scientific issue, is that the tox studies don't actually support the proposed dose. So the rat study showed no significant toxicity, but that actually wasn't a good thing. There were questions about exposure, there were questions about metabolism, there was something a little worrying about that study. The dog study showed clear toxicity and death, and it was not completely dose-related. There was toxicity at multiple dose levels, which is normal, but they were not necessarily what you would expect to see a clear correlation with severity and dose. However, there was an HNSTD, and the dog could support a dose of about 40 mg. So this was sent. The sponsor agreed to lower the dose. So that problem was also resolved. However, that problem led to a new problem, is that the issue that their lowest formulation that they had available was 75 milligrams. And so they needed to generate additional CMC data to support the product being used at the lower 40 mg dose. My second example is a biologic. We'll call it Bio 101. It's a bispecific targeting CD3 and a target expressed on non-hematologic cells. The sponsor submitted an IND with no toxicology data and really no primary pharmacology data, although they had some literature references. And they also had some comparisons to an antibody with the same co-target, so not CD3, but the other target. This one went on immediate hold, and the citation for this hold was 21 CFR 312.42b, insufficient information to assess risk to human subjects. There were hold comments from all disciplines, but the major comments from non-clinical were to justify the lack of toxicology data or provide a GLP-compliant 28-day toxicology study. If there is no relevant species, provide human tissue cross-reactivity data and provide pharmacology data to support the dose, because this one was going to need a MABEL-based approach to a dose because it's an immune agonist to support that supporting the appropriate starting dose. So the sponsor submitted a complete response. They submitted data showing that there was no relevant species. The product bound to the co-target in some species, but not to the CD3 epitope in any species except chimpanzee, and we're not going to ask for a chimpanzee study. There was no GLP-compliant tissue cross study, but they did do a preliminary screen, and there was information on expression of the co-target that they provided, and FDA determined that this was acceptable. And finally, the sponsor included some dose comparisons to traditional antibodies with the same co-target and to a cellular therapy. They also provided some proof of concept data that included humanized mouse models. So the outcomes of this, FDA still did not agree that the available data supported the proposed dose. The comparative data with the vastly different kinds of products, we didn't think the comparisons were valid. There was no evidence of there was evidence of in vivo cytokine release and death in the proof of concept studies that they provided, which was very concerning. The argument for a safety margin based on the lack of PBMC response in the presence of normal cells was not compelling, particularly because the co-target was widely expressed. So they put these PBMCs in the presence of cells that don't express the target, and we're trying to say that that provided a safety margin. But again, because the target was so widely expressed, that was not an argument that was all that believable. There was, however, sufficient in vitro data to support a dose. The EC50s in the activation assays were in the range of 10 to the -4th nanomolars, and at a dose of approximately 112 nanograms, the predicted Cmax for Bio-101 would be in that range. But this dose was about 100-fold lower than the proposed start dose of 11 micrograms. And ultimately, the sponsor remained on hold because of a lack of sufficient CMC data regarding the ability to adequately deliver the dose of 112 nanograms. This has been a problem with some of the CD3 bispecifics, and in particular because some of them require very low doses, and at the starting dose, they can absorb to the infusion apparatus. So that people were not actually getting the initial dose levels, and they would get to a point where they stopped absorbing, or they had enough drug that they weren't absorbing it all to the sides, infusing people, and they were getting too high of a dose, and there were safety issues. Now, after seeing all these two case studies, I just wanted to emphasize that it's not actually our goal to find a reason to put your study on hold. We really try to work with people to avoid holds and answer any questions that we can. So, final summary to remember is that there is a lot of non-clinical data needed to open an IND. You should go to S9 as your starting point, invest in good GLP studies, and consider your non-clinical data before finalizing the initial clinical formulation. And now Dr. Donahue is going to tell you more about the clinical considerations for a first-in-human protocol, and I'll be back later for questions. So thank you for your attention.
Hi, Whitney. Thank you so much for that talk. It was distilled conciseness. Well, I think we're gonna ask you questions before we hear from Martha. Well, there must be some gaps in our guidance documents, because the same questions come up all the time. And one of those is, maybe it's because oncology is trying to be flexible, but do GLP studies have to be done using GMP material?
Hi. No, can you hear me okay? Sorry. Okay, I'm sorry. Yeah, you don't have to use the GMP material or the clinical batch, but you do need to use a batch that's made using the same process. Often we get a dirtier batch to start with for these small molecule drugs, especially to help qualify impurities. I think Olin and Teresa talked about that earlier today. And in oncology, in our space, we do have some flexibility around impurity qualification. You don't necessarily have to fully qualify the impurities. at the maximum dose that you think you're going to reach, but especially at your starting dose, we want at least some impurity qualification. So that's an important consideration.
Thanks. The other question that we always see are combination tox studies required for your investigational drug and an approved anti-cancer therapy?
Yeah, this was a topic that actually was included in the ICES9 Q&A document, and we don't generally require combination toxicology studies unless in certain situations where you have like an enhancer drug that it's a PK interaction to make one work. In that case, you might have to do a combination tox study. But generally speaking, that is not something that we're looking for. We're looking for the tox profile of each individual drug. Where we will maybe ask for some additional studies is some pharmacology studies, just looking for some like basic synergistic very broad endpoints. So you're looking at mortality, you're looking at like massive weight loss in these studies, but you're not doing like a full tox study. And for a lot of the IO products, you're looking for maybe whether or not you're seeing a big synergistic increase in like cytokine upregulation or something like that to help support using the combination, but still not an actual full tox study for that combination.
Great, thanks. Here's a question: Would the ERG assay for oncology indications have to be GLP, similar to for a non-oncology indication, or would a non-GLP screen ERG assay be sufficient?
So generally speaking, if you're going to do these safety pharmacology studies, you do want to do the GLP study, and that's going to be for the ERG assay or for whatever kinds of studies that need to be GLP should probably go ahead and be GLP. We're not going to ask for these for a biologic product, these large molecules. That's not something that we're looking for. And technically, the ERG assay is usually done during screening, so we see it early, but it's not technically required. You just need to have some sort of cardiovascular assessment. If you have cardiovascular assessments that are built into your tox studies and you haven't done the ERG, that's probably not going to be a hold issue for oncology. But because it is kind of used as a screening assay, it's something that is often included for these small molecule drugs very early in development.
Thank you. I'm going to read this one. I know the answer, you know the answer, but I just want to restate this. For a biologic, the early proof of concept non-clinical studies utilize research-grade material. Does FDA want to see any comparability data to the proposed clinical material in the IND filing for the previous proof of concept trials or studies?
That's not stuff that we're looking for in your pharmacology study, your proof of concept. That's stuff that we're looking for in your toxicology study usually. So no, I would say the answer to that.
Thank you. And then this one, it's an interesting question. I think I'm assuming these are hematopoietic-induced pluripotent stem cells. Can they be used instead of primary human cells for cytokine release assays? And I'm guessing there might be some questions on how well you differentiate those into the appropriate, hematological cell lines for that to be useful. But let me hear what you have to say.
Yeah, this is an interesting question. I don't think I've ever seen anyone do this. I mean, your primary, your PSCs aren't necessarily going to make a lot of cytokines. So I don't, if I were planning the study, I would not say that was a method that I would recommend. Primary human cells are relatively easy to get for cytokine release assays. My undergrad or my graduate school advisor used to tell me, and I think that it would be. I don't know that that's a method I would try to use at this point.
Yeah, it seemed kind of complicated to me as well. One last question before we have to go, and that's with regards to a MABEL approach. I know it's know your drug, know your assays, but are there certain obvious pharmacology studies that one should do for various products for the MABEL approach?
Well, other than doing your cytokine release assays, I think what we're really looking for in those is concentration response data. And the cytokine release assays can, if they're negative, they can help you maybe support a higher dose. And this is part, I think we got a lot of questions about the MABEL and how we're balancing safety and efficacy. This is an issue for oncology. We don't want to dose so low that the patients aren't benefiting, but with these IO products, especially, some of the animal models are not convincingly representative. So when you have that situation, we kind of tend to go low and slow. We're looking for maybe a mixed lymphocyte reaction to look for activity, and you're looking at a concentration response in those assays, and maybe you can support a slightly higher dose if you're not having massive cytokine release reactions in your other assays. So those studies are very important, and in that situation, you definitely have to submit all of your pharmacology data and not just like summaries of your pharmacology data. So for most of the biologics, especially, you want to submit that primary data. And I think that the other way that we kind of try to address that is by allowing for more rapid dose escalation and smaller cohort sizes to try, and intrapatient dosing to sort of help to get you to a place where you're actually benefiting patients, but still at a dose level to start with that we think we can support based on your data.
Thank you. Yeah, it's always impressive to me that we use a dose of micrograms for some of these bispecific engagers. So, Dr. Helms, you have addressed those questions with aplomb. Thank you so much for taking the time to be here today. And I think I saw Martha enter our virtual forum. And Lisa, I'm going to hand this over.